So I'm going to be just drilling down with a walkthrough of our search and navigation features, just to give a more detailed introduction to those capabilities. Before I do, we like to give you a sense of the organization of data in FaceSpace, because it's kind of good to have at least a rough idea of what kind of information is in FaceSpace besides the actual data files that are in FaceSpace, so that you have an idea, some intuition about how to navigate around for data of interest. So we have this kind of high level schematic here on the right-hand side of the slide. And I'll just walk through it because this gives a high level picture of how our data are organized. So starting from the top, we have a concept of a project, which is, as you would expect, either a team of researchers, a lab, a collaboration of researchers who are producing data. They contribute data sets. A data set is just an overall collection of data and metadata. If you're not familiar with the term metadata, it just means data that describes other data. So in FaceSpace, we not only collect and distribute, say, the images as a TIFF file or the omics data as FASTQ files, but we also want to collect enough descriptive information about that data so that you understand the context in which it was collected. So in our data collection, so experiments are records of the procedures, like particular types of assays, a ChIP-seq or RNA-seq or single cell or micro-CT or other type of assay that was taken on a biosample. They should follow and do follow protocols. And we have a way of recording protocols and associating them with experiments. And then those procedures are performed on some form of a biosample. This next part, we're working through this next part. It could be a little bit confusing to see both of these terms used, where we have the term replicate and biosample, and as Chris mentioned, we were working on some usability improvements. So over time, I hope that this will be more intuitive as you walk up to it. But, let me just try to explain. So we record the biological characteristics in one area of the database called the biosample. So that's a record of the species of the specimen, the maybe the development or age stage, the genotype, and so on. A replicate, the record of a replicate, is really just used to associate a biosample with an experiment. So you might think of a particular specimen taken from a mouse at a particular time point is just that. It's a specimen. Until it's used in an experiment, it's just some sample that exists that was taken. Within the context of an experiment, then you would think of having this is a set of biological replicates. You might have 3 samples taken to be the control and 3 to be a particular mutant group in an experiment. So those then show up as the replicates that are associated with experiments. And then the data, again, I mentioned there's imaging data, omics data, other forms of data, are then associated with a specific replicate of a specific experiment that is associated with has a protocol. So you can get a sense quickly of where the data come from, what's the context of it, what was it taken from, and all of that drives the search and navigation within FaceSpace. So our overall goal with the data that we collect and distribute is that we want to provide you with data that is suitable for reuse, for reproducing results, and for analysis or reanalysis. So the data that, by and large, almost entirely that you'll find, if it's not reference data, what you'll find are the highest resolution, the earliest data that we could collect from those who contributed data to FaceSpace. So not just, say, figures or image panels or diagrams, but the actual reconstructed CT image, for example. So we want to get the higher resolution data, not just, say, the things that show up in a publication. So That's the kind of data and the kind of description of it that we need to help facilitate that usage and reuse of the data. So I want to just stop for a moment and provide a little analogy, because we get this question about finding data in FaceSpace and a comparison to, say, Google or PubMed search. And I think that a useful analogy is if you imagine shopping for a car online. If you were looking for, because in that mode, you're not just looking for a particular document, but really pinpointing something very precise that you want or need. So if you were to look for, say, a 2018 or newer Mazda MX-5 Miata with less than 20,000 miles near 90292, and you could try that for yourself in Google, and you'll probably get some results for sure that are relevant to Miatas and certain location and maybe certain years. But you'll have a haystack to look through. And you can contrast that with a different style of navigation that we see on the right-hand side. And I'll just provide one more example of that, which, there are many such sites, but you could see cars.com is one particular example where this mode focuses on different specific filters that you pick from the characteristics of the item that you're looking for. So that tends to work better when what you're looking for is not a text document, which is better for like the PubMed or Google Scholar kind of searches. So this mode of navigation is called a faceted navigation. And as I mentioned, it basically focuses on filters by different specific categories of information. So again, with the car shopping analogy, you have something like the year, make, model, mileage, distance from where you're at, because you have to physically reach the product in this case. And then, if you were to go to cars.com, you won't see a search box on the homepage. You'll see on the left-hand side, you'll see a variety of little select filters that they've chosen to kind of give you a jumping off point. And then on the right-hand side, you get the actual search listing. And again, you see these filters, some that are selected, you have new options, you can expand some that you've already picked from, you can pick from new categories, and so on. So this is the mode of navigation that we have adopted in FaceSpace. And it's actually quite common. It's not the Google or PubMed style, but it's actually very common on the internet for finding and pinpointing specific exact types of data. And so I'll just kind of proceed to the website at this point and do a little tour of these features. But just as an overview here, on the left-hand side, we have that home page. And again, you won't find a search box, but you will find a set of kind of a simplified set of these categories and filters. And these are not exhaustive by any means, but they are just some of the more common ones that have been identified by our user base. And then on the right-hand side, you kind of drop into a search or search results and have, again, the set of filters that you can expand or choose from new ones. All right, so with that, let me just switch to the website. And okay, so let's walk through these. So again, here we are at the home page of the site. We have, again, here a set of, these are just kind of shortcuts to picking from the filters that we have in the overall search results experience. And then we have a box here, search all data sets. As Chris mentioned, we have a nav bar so you can go to different categories of information. And again, like the diagram that I showed there at the start there, you're going to see those, again, data sets, experiments, biosamples, projects. Those are some of the key entities in the system. And then we've added images, although we have, of course, other types of data. Image search has been something that has been specifically requested, and so we've added that interface that Chris highlighted. Chris also showed the mouse data summary. Again, although we have other model organisms than mouse, we have a good collection of zebrafish data, human data, and a little other data, as well as interest in any of you who are doing research on other animal models and would like to contribute. So as Chris mentioned, we have this interface. it does scroll to the right. So it's not done there with what we can see. There's actually a lot more over to the side, and everything is actually drills down into that faceted navigation. So again, this is just another way of accessing that. So all of these links along the top are active filters to get you information on the anatomy along the top, the age stages down the left, and then the experiment types. And then each of the little boxes are clickable. So each one, you don't have to click the little color band. It's not quite that precise, and those get pretty tiny, but that's more to show you that there's a variety of experiment types, which are color coded along the bottom for just a quick view. And any of these will drop you right down into the same search, faceted search interface. So I'm just going to go ahead and start from the top and pick mouse. So this is going to drop me into the search results here for just for mouse. So I have relatively few filters set. Only one set is really for species. So we see down here on the left-hand side, we have the species category of filters here. You can see I can expand it to include zebrafish, human, chimp data as well. And then you'll see at the top, we have experiment type. Now we have a set of experiment types that are visible here. So if I was interested in fluorescence microscopy, I can see it right away. I can see that there would be 4 matches. I'm going to uncheck that for a moment. You'll also see there's a link here for show more. So what we see here are not the complete list. So let's expand this and see what else are available. Okay, so there are actually 37 different experiment types available. And we also see more going on here. So these are terms drawn from vocabulary produced by the informatics community. So these are, wherever possible, these are precise terminology. So you'll see they have their own accession numbers. These are not face-based accessions, but external terms. So to the extent that we can make data interoperable, we want to adopt any terms that are available. And when they're not, when it's internal term, you will see a face-based accession there. But you have the names, we have descriptions, so we can sift through this to find information that we would like. So like, let's say I just want to see imaging, a variety of imaging assays. So I can check that, submit, and there it sets the filter here on the left side too. And now I've filtered my overall result set to match 553 results. Now, along the left-hand side, as we go down, we will also see that there are a number of other little labels here, and these all expand. So if I'm interested in a particular age stage, let's expand that. If I click on that, I will get the age stages. Again, if I'm wondering if there's more than what I see in here, there's always show more. I click on that. I can see there's actually 31 age stages that I could choose from. And so on down this interface. So I could do the same thing for anatomy. I can close them and so on down the list here. Okay, so the next thing you would see is at the top we have, or near the top here, we have a couple boxes with little X's next to them. So I've got the experiment type, imaging assay set. So this tells me that this one is set already. So let's just say if I had set a lot, I might not want to find the ones I've set down the left-hand side. Like, let me just choose a few of these here. Okay, so let's add a few of these age stages. So now, let's say I don't want to locate those. I just want to clear the filter for age stages. So I've got 144 matching results for these stages, and I can just click the little X, get rid of them, and essentially back up to the point I was at. Another nice little feature over here is this permalink. So if I click it, you copy that to your clipboard automatically. So this is useful if, say, you wanted to share, you found a search result here that you either wanted to bookmark for yourself, for your own reference, copy it to some notes, or share it with someone. The permalink is a good way to do that. And sometimes it can be more reliable than copying and pasting out of the browser toolbar, because browsers sometimes change things up or add spaces or add some encoding. So they're not all are always consistent. So the permalink is a really good consistent way of always being able to filter. We do have a little bit of free text search going on in the interface. So I did want to mention this here at the top, right under the word data set. So you can do a certain amount of searching. So again, most of our information is structured and there's an organization to it. But there are titles and descriptions in the data sets. So if you wanted to search across those, like let's just say, okay, molar. Let's see if anyone has molar in the title or the description. So here we appear to have that. So I can see right here, let's see. Okay, we've got molar in the title here. I don't see molar here, so I bet it's down in the description. So you can get some refinement if you're, if you already know that there's a certain data set of interest and you know some words that are in the description. But the most effective way is to look through these filters down the left-hand side. Okay, so now if I just drill down, let's just see what we get here with this first record. Oh, and the other thing is we have a couple links here. So this little view link and this record ID link, these will take us to the same place here. So the record ID is kind of our internal identifier. So some repositories call these accessions, but that's not necessarily a common term across the board for everyone. But essentially a record identifier or this view detail. So either one will take us into this data set. And then what we get when we drill down is that we have the title of the data set, we have a description, and I just want to point out this identifier section. So this gives us the official face-based record identifier. This, what we call the accession, is actually, it is a somewhat de-emphasized identifier in face-based these days. So we keep it around more for historical purposes. Our new identifier system lets us identify everything throughout face-based in a more consistent and persistent way. So we generally work with these identifiers when we're just referring to something. And then data sets and projects have digital object identifiers. So we go out to one of the data DOI registers, and we go and we register the data sets and the projects there. And so these provide a link through the same resolver system that gives you archive grade identifiers for journal articles. And then let's just kind of keep going down. So with every data set, we have the attribution of it. So it was contributed by this particular project here, the development and validation of novel amylogenesis models. We have a couple contributors for it. So these are like, in some sense, the authors of this data set. So when it comes to citing a data set, which will be discussed again later, there's you have that information up front. So I'll leave that for later, but I just want to mention it here in place. And then we have the protocols that are used, and these are actually specifically referenced from the experiments within this data set, but we highlight them at the top. So you get the initial summary data at the top here. And all of these actually are links that will take you off to these terms and stuff. So if you wanted to kind of navigate around face-based and see, well, what else is there for postnatal 7 weeks? You can follow that link and go there. If you're wondering more information about these, are there any descriptions? You'll notice on the right-hand side, there's a table mode. So if I click that, I get an expanded view of this section. So now I see the official OBI, that's the Ontology of Biomedical Investigations. I see their specific identifier, the preferred name for that term, and then I see the description that's coming from that, that vocabulary system. Okay, so that's a way of just kind of looking in, and not all will have descriptions, but many will. The next thing that we see are thumbnails here, and then as we go down, I'm not sure why some of those weren't viewable. So what we have next here are the experiments. So again, we have the data sets organized by experiment. So we first have experiments, and then with them, they have a number of biosamples that were used in them. Again, when you see the biosample and replicate show up, it's really referring to the same thing, but the replicate is just the association of a sample to an experiment. Then we get high-level overview of each of these experiments. And if I go down to the biosample section, then we'll see the, again, just all data set wide, all of the biosamples that are used. But the best way to really know what's used in an experiment is you drill down into the experiment, which I'll back up and do next. Then we see there's sequencing data available, and then imaging data. On the left-hand side, you'll see a table of contents, so we can jump around, so we can jump back to the experiment. Okay, so if I go up here, I can then directly drill down. So let's look at one that has, let's see, which one? Okay, let's look at this one, because this will have interesting imaging data, H&E experiment. So I drill down here. All right, so now I'm in an H&E experiment. I see that I have 3 protocols that are associated with it. So we can take a look at those protocols, learn more about them. We can see the replicate records for this, and then we see the imaging data, and we can drill down here to get, if we want to kind of get a quick preview of this data. There we go. And I'll mention here that one of the new features of FaceBase that we recently rolled out is a new imaging pipeline. So all of our imaging data are processed in a new automated pipeline. It covers most of the data. You know, there are sometimes issues with formatting or encoding of data that we're still working on getting complete coverage, but we have pretty wide coverage of our data. It covers TIFF, it covers Nifty, even other proprietary formats can be processed in it. It generates the thumbnails. So this one is actually a user supplied thumbnail. So if the contributor of the data set provided a thumbnail, we favor that one. If not, we always attempt to generate a thumbnail, and then we'll display that if the original contributor didn't provide a thumbnail. And then also that online viewer, we process the data. The data that you download is the original data contributed, but we process that data for a efficient online browsing experience. And I'll give a couple more examples of that. But I just want to back out here. So we're looking at that experiment. So another thing you'll see is that under an experiment, you have the link back to the data set. So you can always kind of get back to the containing data set where this experiment came from. I'm just going to back up a step because I just want to show this last feature here. So let me drop down to the imaging data. So I just use the table of contents, and you'll notice there's a little explore link on the right-hand side. So if I do this, I'm going to actually drop into a imaging search experience that has already been filtered just for this data set. And now I can apply new filters. So I can come over here and I can say, well, let's see, what do you have in terms of this, the H&E data? Okay, so I just filtered out among all of the data in the data set, one particular experiment type, and I can do that on down the list. I could say, give me the postnatal day 14, and here we go, and so forth. So you basically can, and you can navigate around the experiments and the biosamples and any of the data that are in the data set in these different sections. You can continue to search within a data set by going to any of these. Let me just drop down here, any of these explore buttons, and they're on each of these particular sections of biosamples. Sequencing, well, we see we have just one record, so we don't need to go further down. Okay, so before I hand this over, let me just give a couple other examples of some of the online visualization that we have. So over here, I just have these queued up. So in this particular data set, this is, let's take a look for it right here, RNA-seq assay. So that's the kind of experiments that are here. And the contributor of this data set provided gene annotation tracks. So we have embedded browser from the UCSC genome browser window. So we can look at the data, get a quick view of it. We can, you can do it actually in the embedded window, or you could click this full screen view and you'll load this in its own window. And then you can just use the genome browser interface as you normally would. So of course, our default view of it is probably not right where you want to be looking at it. And you can use its interface to navigate around. Let me go to the next data set here. So in this one, okay, we have tooth root development. We have some fluorescence microscopy. We have micro CT data. So I just want to show we do have this OrthoSlice viewer available on the site. So in a number of cases, so we don't have this fully automated. So the coverage isn't as complete as the other pipelines. We'll be working on getting this more automated over the coming year, but a number of data sets have this as well. And you can kind of just move through the image in different orthogonal angles here. You can also switch into a 3D mode. Not everyone knows this exists. It's over here on the side panel. And then you can get this little quasi 3D view that is rendered in the browser. Now, if I did this in real time, you would see it say processing about 3 times. And then after that, it's you're off. You're fine from that point. No more delays. Okay, and then the next one, this comes out of our new imaging pipeline. Again, one of these images. So this was, I'm sure, a TIFF image that was processed. And you can see that we can zoom in on it, and we can really go to whatever maximum resolution was provided by those that contributed the data to the data to FaceSpace. Okay. And then I just want to give one more example of that viewer. So if an image has a large Z stack, so we can navigate through that Z stack. And you can just walk through all of the series of images. There's also a panel available that lets you play around with some of the settings. So let's see. If I take this, yeah, so all of this is in browser. We can manipulate some of just a few of the image settings here to get a better look at it. Okay, and then as Chris mentioned, we have this imaging search now under the nav bar. You can get to it, data browser, images, and this pretty much just allows you to navigate in the same way that I showed, but over the image data. And many of these thumbnails are now generated, and all of the underlying high resolution views of this are generated now by the pipeline. So yeah, with that, that concludes my tour. And if you have any questions, I can take those now before we move on to the next presenter.